Expression of programmed cell death receptor 1, PG1, and its ligand, PGL1, on immune cells and astrocytes is elevated in cerebral cortex in a mouse model of Alzheimer's disease by Natalie Kozyrev, Arjun Rylett, and Gregory A. Dekaban. It is critical to examine the role of the innate immune system in order to identify when different immune cells become mobilized in response to the accumulation of A beta plaques. Some evidence exists to support the idea that in the course of Alzheimer's disease, monocyte-derived macrophages invade the brain from the periphery and promote an immunosuppressive effect on resident microglia and microglial-derived macrophages. This results in a halt of phagocytic activity and a disruption in A beta clearance. The immunological dysfunction observed in the Alzheimer's disease brain may be the result of microglial and hematogenic macrophages entering an exhausted or frustrated state. Exhausted, frustrated macrophages express cell surface molecules called immune checkpoint proteins, including programmed cell death 1 or PG1 and its ligand PGL1. In the current study, we investigated the spatial and temporal localization and expression of PG1 and PGL1 in the brain. We used lysozyme EGFP knocking transgenic mice that express enhanced green fluorescent protein, or EGFP, in hematogenous but not in microglial macrophages, enabling discrimination between the two cell types. The lysozyme EGFP knocking mice were crossed with 5XFAD mice, and the offspring rapidly developed Alzheimer's disease pathology, including A beta plaques, gliosis, and behavioral deficits by 5 months of age. At 3 months of age, we observed significant increases in the area of PG1 immunoreactivity in the cortex in L5F plus females compared to age-matched L5F plus male mice in controls. At 5 months of age, we observed significant increases in PG1 and PGL1 area of immunoreactivity in the cortex in both male and female L5F plus mice compared to controls. At 7 months of age, we observed significant increases in the area of PG1 immunoreactivity in the cortex in male and female L5F plus mice compared to controls. In addition, we observed significant increases in the area of PG1 immunoreactivity in the hippocampus in female compared to male L5F plus mice and controls. Brain sections from a 7-month-old L5F plus female mouse containing the cortex were triple immunostained for anti-EGFP, a marker of myelomonocytic cells but not microglia, anti-IBA1, a macrophage and microglial marker, and either PG1 or PGL1. In a separate experiment, sections were triple immunostained for anti-EGFP, anti-GFAP, a marker of astrocytes, and either PG1 or PGL1. Preliminary assessments revealed that PG1 is expressed in hematogenous and other EGFP-positive cells in the cortex, while PGL1 is expressed on hematogenous as well as microglial macrophages, astrocytes, and EGFP-positive cells. A temporal comparison revealed that 5 months represents the age when both PG1 and PGL1 peak in the cortex in male and female L5F plus mice. Future studies will examine the different cell types in greater detail in order to determine which cells more highly express PG1 or PGL1.